Now to continue from the last video. Last video we focused on glycolysis. Key thing here, we're going to talk about high energy carriers. In photosynthesis we talked about NADPH. Big thing here, that P, think of photosynthesis. Because respiration has a similar looking one of just NADH. As well as another one called FADH2. So two energy carriers, photosynthesis had one. Look for that P is the one in photosynthesis, the other two are in respiration. The key thing with these two is to bring electrons to step 3. Advantages of glycolysis. Remember, this means splitting sugars. So it is a very fast process because it's simple. You're going from glucose, which has 6 carbons, to pyruvate, and you are forming 2 3-carbon molecules. Another advantage is this can happen when there is no oxygen. We call that anaerobic respiration. So the first step in any type of respiration is glycolysis. If oxygen is present, it goes one pathway. Not present, goes in the other. The downside is anaerobic provides very little energy. Oops, spelled anaerobic wrong. So anaerobic provides very little energy as a result. Only 2 ATP, because that is what glycolysis produces. That's it. So it doesn't go to the other 3 steps, or the other 2 steps, because you need oxygen in order for that to happen. So anaerobic only has 2 instead of getting the total 38. Now step 2 is the Krebs cycle. The Krebs cycle is more of a prep stage. This is going to be similar to the light reaction of photosynthesis, where you are getting things ready for the next step. We are in the matrix of the mitochondria, that space within the second membrane. This is where the DNA and the ribosomes are of the mitochondria, making it a very unique organelle in addition to its 2-membrane status. We are starting with 2-pyruvate because that is the end of glycolysis. So 1 pyruvate at a time. So the 3-carbon molecule becomes a 2-carbon molecule, and that carbon eventually is released as CO2. Through a series of reactions, it goes through a cycle. And within that cycle, we lose kind of the other 2 carbons as CO2. So 3 carbons lost per pyruvate makes sense, it's a 3-carbon molecule. This happens twice. Why? Because we have 2-pyruvate. And each spin around the cycle, each turn, gives you 1 ATP. Since we have 2 turns, it ends up with 2. Now, the main important thing here is we are building up your NAD and FAD. Quite a few of them each step of the way. So we're getting things ready for the big step 3. Getting those high energy carriers prepped to unload in step 3 to make all of our ATP. Or majority of our ATP, should I say. So 2 turns, 1 for each pyruvate. The total amount of things produced is, remember, we are unleashing 6 of these. Remember our reaction for photosynthesis, 3 at each turn. But more importantly, what are we gaining? 2 ATP, 1 for each turn, 8 NAD, and 2 FAD. NAD and FAD, the energy carriers of respiration. Getting things ready for step 3. So CO2 is released, ATP is used up, because it requires energy to kind of move stuff, kind of prolonging into step 3. And then NAD and fat are being taken to step 3, which is the electron transport chain. And that's going to be our big money step to getting majority of our ATP, making it the most efficient step of respiration. So where does this occur? The key word here is mitochondria. If you understand that, I'm cool with that. But if you want to get specific, it is in the space inside the second membrane, the matrix. If you're in the mitochondria, that means oxygen is involved. Oxygen is like the ticket to get in the mitochondria. What to begin with? Your 2-pyruvate. Remember, each of them are 3 carbons. And since we have 6 carbons here, remember we have 6 carbon dioxide, they're going to be released. So 6 CO2 is released. We have 2 ATP formed. So that's kind of a waste product, so I'll kind of separate that out. We have our 8 NAD and then our 2 fat. I'm not terribly concerned with the numbers of your NAD and FAD, just understanding that this is a big step in terms of getting your high energy electrons and whatnot all prepped, ready to go in these molecules. So your electrons, your hydrogens, getting things ready to go for step 3. So what happens to each of these products? Well, carbon dioxide is released. This is going to be used up. And these are going to be transported to step 3. So far, everything, this is in the cytoplasm. Glycolysis, if oxygen is present, then we enter the mitochondria and we stay there until the end. So what we have here, step 1, if there is oxygen, we enter the mitochondria. If there is no oxygen, we enter what's called anaerobic. This is going to be a type of fermentation we talk about later. The Krebs cycle, we are releasing our carbon dioxide, getting your NAD and fat all prepped up, and then going to step 3, which is when we are involving oxygen and when we are forming water. From a big picture standpoint, we're going to take a step back. Prokaryotes do not have mitochondria. So that's not where respiration occurs. Respiration occurs for them in the cytoplasm. Remember, all cells have 4 basic components: cytoplasm, cell membrane, genetic material, and ribosomes. Since they're very basic, their metabolic reactions are happening in the cytoplasm. Eukaryotic cells basically can have 2 things happen. Aerobic respiration, which means oxygen is involved, is in the mitochondria. If there is no oxygen after glycolysis, we stay in the cytoplasm. And we'll talk about anaerobic in a few minutes. So stage 3, the electron transport chain. This is complicated. So we are, once again, in the mitochondria, specifically in the crista. So when you look at a basic mitochondria, those folds, that's called the crista. NAD and fat are going to be stripped of their high energy electrons. So here's what happens. Think of this as a race. And those electrons have different checkpoints to go to. And when you have a flow of electrons, it creates a little bit of a current, a buzz. And that buzz is used to form energy to power something else. Now, what it is going to power is the movement of hydrogen ions across or should we say against the gradient. We have a low concentration here. We have a high concentration here. So we need energy for active transport. So your electrons, as they hit each checkpoint, are creating energy to allow this movement to happen, going against the gradient. So hydrogen ions being transported out into the mitochondrial space. Now, when they do that, they will eventually head toward the ATP synthase and come back in. The movement of those, the movement of those hydrogen ions is harnessed by ATP synthase, because that's creating a little bit of energy to then build ATP. And that's where we get our 34 ATP made. However, there is one important extremely major detail, is the finish line of this electron flow is what's called the final electron acceptor. And that is oxygen. Oxygen will accept electrons and combine with two hydrogen ions to form water. So the final electron acceptor is oxygen. Now here is the important thing. If this is blocked, and this is what cyanide does, if it blocks the formation of water, it blocks oxygen from being the electron acceptor, then the ATP synthase step does not happen. So oxygen needs to form water before ATP is made. So it has a crucial role in making sure ATP is being made within this third and final step. So essentially, electrons moving down, combine with oxygen and hydrogen ions, because two electrons, negative charge, two hydrogen ions, positive charge, that cancels out to form a neutral water compound that is released. When water is made, then ATP is formed. And how much are we forming? 34. So 34 of our 38 ATP is in step 3. Quite a bit. So where is this located? Mitochondria. If you simply understand that, it's fine by me, but if you want to get specific, it is in those folds called the crista. What do they drop off? High energy electrons. Because the electron movement is what causes the buzz for the active transport to happen with your hydrogen ions. Your electrons will then bond or combine with oxygen as well as your hydrogen to form water. When water is formed, then your hydrogen ions will go through ATP synthase. That energy will be harnessed from the movement to create ATP. So ATP synthase, once again, our maker, or should we say our builder of ATP. And your H plus ions are going to flow through. So most important role here is oxygen as the final electron acceptor. If its role is compromised, then this ATP is not made and ATP synthase cannot do its job. So real quick, the movement of high energy electrons allow for the production of ATP. As the electrons move down the chain, think of it as a race with different checkpoints and a finish line. Energy is released because movement of electrons creates a current. That current is harnessed to move your H plus ions across the membrane. This is active transport. That's why we need energy. The ions then rush back, because that's more of a natural thing, across the membrane, producing enough force to spin the ATP synthase and generate an enormous amount. So when you're moving things kind of against the will, you're creating a little bit of a chaos. And they want to revert back. And that movement coming back, that energy is harnessed to add a phosphate to ADP to make ATP, specifically 34 of them. So of our steps, we are in the cytoplasm with step 1. If there is oxygen, we are coming into the mitochondria, and that makes it aerobic. So step 2, remember this happens 2 times, very little energy in comparison, and then essentially our NAD and FAD, extremely important in delivering your electrons to the electron transport chain and kind of get them moving. We are focusing on the electron transport chain, developing, or should we say forming 34 ATP. So we got 2, 2, and 34. I like to write the Krebs cycle as like a 1 plus 1, just to remind yourself that this happens 2 times since we have 2 pyruvate. But 38 ATP per glucose. So when we look at the reaction in its entirety, glycolysis breaks down glucose to 2 pyruvate. We don't really see oxygen until step 3 as the final electron acceptor. And just as with photosynthesis, oxygen and water are connected. This becomes water whenever it combines with 2 electrons and 2 H plus ions. And when this happens, then our 34 ATP happens. In step 2, carbon dioxide is released in 2 different turns, 3 and 3, to help kind of build up our NAD and FAD. Just as we stated before, oxygen and oxygen and water in the same step, and we see ATP made in every step. But most efficient step is step 3. That is where the ATP kind of treasure is, getting through step 3. So the importance of respiration, well, glucose is kind of a storage. We need usable energy, and that's why we form ATP. Now, the importance of aerobic is just how much it provides. However, if there is no oxygen, we get into anaerobic. Anaerobic is also known as fermentation. And if there is no oxygen, we stay in the cytoplasm. If there is oxygen, we go to the mitochondria. So oxygen is the ticket. Now, if we stay in the cytoplasm, we just stay where glycolysis is. Glycolysis is in the cytoplasm. The pathways are not very efficient because regardless of what's happening, we are breaking one glucose, anaerobic or aerobic. The glucose amount starting does not change. So what happens is you are relying only on the 2 ATP made from glycolysis. So anaerobic only gives you 2 ATP, and that's the 2 ATP from glycolysis. And that is it. Period. 1 glucose, 2 ATP versus aerobic, 1 glucose, 38 ATP. Not very good in comparison. So the 2 types, alcoholic and lactic acid. We're going to see lactic acid with two things, exercise as well as some of our foods, and we'll focus on foods with alcoholic. So with alcoholic, our big example is yeast. Yeast is involved in making bread as well as wine and beer, all your alcohols. Essentially, if we are making bread, what is happening? Bread is rising because of CO2 being made. And why we don't taste alcohol in bread is because it is evaporated out when we bake bread. Another key thing is we're making the lower energy form of NAD, NAD+. So how long this reaction is going to be based off of how much NAD is produced. So bread's an alcoholic, yeast being our big example. So what's happening here is we have glycolysis, step 1. There is no oxygen, so we trigger alcoholic fermentation. What that does is it creates alcohol and carbon dioxide. And then in glycolysis, as we stated before in the first video, we are making NAD and then breaking that down. And we're going to be limited by how much of the NAD plus before that reaction stops, and we have to kind of revert back. So all this happening in the cytoplasm because of no oxygen. Lactic acid, similar, no oxygen, so we stay in the cytoplasm again. So step 1 gives us 2 ATP, and that's all we're getting. We have our 2-pyruvate after glycolysis, and that simply is just going to break down into lactic acid and NAD+. And your lactic acid is also known as the burn. When you're strenuous exercise and you start to feel cramping or what feels like acid in your muscles, that is lactic acid. So when your body is not supplying enough oxygen, we get into this secondary form of energy production. It's not efficient at all, and we cannot last very long with it, which is why the burn eventually causes you to stop or you're very tired because you kind of run out. And you have to catch up breathing to trigger back into aerobic respiration. From a food standpoint, cheese and yogurt use the sugar lactose, milk sugar. Lactose is a disaccharide that will break down into glucose. So during exercise, your breathing increases to provide your body with oxygen. However, when your muscles run out of oxygen, too much exercise, very strenuous, you go to your secondary source of energy production with lactic acid fermentation. So you get a little bit of ATP, specifically only 2. And then at some point, you're going to be forced to stop because you're going to get cramping, you're going to be sore, and you're just going to be tired. And when you stop, you're not using as much ATP, and it gives your body time to deal with its oxygen debt, to kind of get breathing going back to where it should and bring things back to normal. So, 2 types of fermentation in review. Kind of pretty basic, alcoholic and lactic acid. Where does it occur? In the cytoplasm. Why? Because there's no oxygen, so we cannot get into the mitochondria. So the energy produced, well, fermentation is an example of anaerobic respiration. We are only getting 2 ATP, and that is specifically from the first step. Aerobic, on the other hand, you are getting 38 ATP. And why that is because you are getting 2 from step 1, 2 from step 2, and then 34 from step 3. And remember, steps 2 and 3 are in the mitochondria because of oxygen kind of being the ticket to get in. So anaerobic only deals with the energy from glycolysis, stays in the cytoplasm. Aerobic, when we get in the mitochondria, if we finish through step 3, we are making the most efficient process to breaking down one glucose molecule. Okay, that is it for cellular respiration. Complicated, but hopefully you pick up some of the big points. We'll keep reviewing. If you have any questions, please ask. Thank you for watching.